All right, let's look at recombinant DNA technology, the general overview. So what is recombinant DNA technology? So as a way of introduction, in the first video, which is introduction to biotechnology, we said in biotechnology, this is a technique or a process where biopharmaceutical products are manufactured using living microorganisms. Okay, so we're saying one of the techniques that are used in the manipulation or in the production of these biological products is recombinant DNA technology, and hence this lecture. So in recombinant DNA technology, we're saying we're using living microorganisms, okay, to produce proteins or to produce biological drugs. So what is this process of recombinant DNA technology, and also what are some of the steps that that are involved to produce these proteins? So this is what this presentation is all about. Okay, so we're saying recombinant DNA technology, it involves the use of enzymes, okay, and various or other laboratory techniques to manipulate and isolate DNA segments of interest. In this process, so this process is used to combine or to splice DNA from different species to create genes with new functions. So we're saying we have DNA, let's say in a human, that codes for insulin, for instance. Then we also have microorganisms, let's say E. coli, that carry DNA, which can that carry DNA, and we can combine our DNA from the human being, which is the gene of interest, and also the DNA of microorganisms. Why microorganisms? They are capable of replicating and hence making many copies of this DNA. So the combination of different DNA species to create new genes, let's say with new functions. So the resulting copies are often referred to as recombinant DNA. So simply, what is recombinant DNA? So it's a process where we use enzymes, to isolate and manipulate DNA. So we combine DNA of the gene of interest and that of microorganisms. Then this product of the DNA that is recombined, or two pieces of DNA that are combined from two different species, is what is referred to as recombinant DNA technology. So this is what is referred to as recombinant DNA technology, simply just recombining pieces of DNA from different species. Okay, so at the center of recombinant DNA technology is another technique which is known as DNA cloning. DNA cloning. So this is at the center of recombinant DNA technology. So DNA cloning, to clone is to simply make a copy, an identical copy of something else. So DNA cloning, so in DNA cloning, what researchers do is that they clone or they make many copies of the DNA of interest, okay, such as, so we'll go with, for example, we'll go with an example of insulin. Okay, so how does this process happen? So I'm saying it involves inserting the target gene into a circular DNA molecule called a plasmid. So there we get it. So first we have the DNA of interest from the human being. Let's say, so we'll go with the insulin gene. And then we have the DNA from microorganisms. What is this special DNA from microorganisms? It is the double-stranded, it is a circular DNA, the double-stranded circular DNA of microorganisms, which is known as the plasmids. So what you do is we identify and isolate the gene or the target, the gene of interest, and then it is inserted into the circular DNA molecule, which is called a plasmid, The plasmid, why plasmids? Because these plasmids can be replicated in bacteria. So when they replicate, it means they make many copies of the gene of interest that they are carrying. So we use plasmids because bacteria can replicate and hence making many copies of the DNA of interest. And in some cases, they can just be used, as storage laboratories for a DNA of interest, or they can be used to express the protein for which they are coding for. So as you can see here, you have a plasmid, which is this state, which is the DNA of interest, and then you also have the gene of interest. So this is the gene that codes, let's say, for insulin. So it is inserted So it is inserted into the plasmid, and then at the end of the day we have the plasmid DNA. We also have the DNA of interest from the human being, and then these two are combined, okay, to make this recombinant DNA. So we have this DNA, okay, from the human being, and then also the plasmid DNA from the microorganism, making what we call recombinant DNA technology. So this is what it's all. Okay, so we're saying in DNA cloning, this is a process of making multiple and identical copies of a particular piece of DNA. Then in a typical DNA cloning procedure, the gene or other DNA fragments of interest, or so this can be medically important human protein, for example, insulin, is first inserted into a circular piece of DNA called a plasmid, as we saw earlier. Then after that, so we're saying the insertion is done using enzymes. So if you remember in the introduction, we say this is a process that uses enzymes and other various laboratory techniques to isolate and manipulate DNA. So how do we isolate the DNA of interest from the human being, or let's say from the human cells, which is the nucleus? And then also, how do we paste it? How is it pasted into the plasmid? So we use special enzymes. So the insertion is done using enzymes that can cut and paste DNA. So these enzymes should be able to cut this DNA from the human cells or from a human being, and then also we have enzymes that should be able to paste it in the plasmid DNA, and it produces the molecule which is known as recombinant DNA, because we have combined two sets of genes or of DNA. Okay, so we're saying after introduction, so after the cutting and the pasting of our DNA of interest into the plasmid DNA, the recombined or the recombinant plasmid is now introduced into the bacteria. So you introduce these plasmid into the bacteria. Why? Because plasmids reside inside bacteria, and that's where they replicate. So it is bacteria that replicates, and as bacteria replicates, they make many copies of these plasmids that are carrying our gene of interest. Then, so we're saying the bacteria carrying the plasmids are selected and grown up. So see what this selection process looks like. As they reproduce, they replicate the plasmid and pass it on to their offspring, making many copies of the DNA of the DNA of interest. Now, you might ask a question: what is the point of making many copies of DNA sequence in a plasmid? So, like I said earlier, uh, in some in some cases, we need many copies of DNA to conduct experiments or to build new plasmids. And then in other cases, the pieces of DNA, they because they could be used because they encode for a useful protein, and the bacteria are used as factories to make proteins. So why are we making copies of DNA? Sometimes we just want factories. The bacteria can act as factories um, to make these proteins. So for instance, the human insulin gene is expressed in E. coli bacteria to make insulin that is used for diabetes. So this is an example that is close to home. What I was saying, the normal insulin that we're using, how is it made? So whether it's a short acting, intermediate acting, and long acting insulin, how are they made? So the insulin gene is isolated from the human being, and then it is cut and pasted into the plasmid DNA of E. coli, which then replicates to many copies of this DNA, and then it is expressed inside these bacteria to make the useful insulin, which is used for treatment of insulin-dependent diabetes. Okay, so let's look at the steps of DNA cloning, and then we'll go into them into details. Okay, so we're saying, let's see how DNA cloning can be used to synthesize proteins such as human insulin in bacteria. So these are just the basic steps. Then later on, we'll look at exactly how is insulin manufactured using recombinant DNA technology. So the first step in DNA cloning or in recombinant DNA technology is identifying and isolating the gene of interest. So what is this gene of interest? The gene that codes for a specific protein that you're interested in. So in this one, for example, if we're interested in manufacturing insulin. So we need to identify and isolate the gene. We need to identify and isolate the gene that codes for insulin. Okay, and then or a complementary DNA coding for the target protein. Okay, and then number 2, you cut open the plasmid and paste the gene. So 1, identify the gene that codes for insulin and isolate it. After you isolate it, now it needs to be pasted into the plasmid. So remember, a plasmid is a circular DNA which is found in bacteria. So first of all, you need to cut this plasmid open, and then you paste this gene of interest in the plasmid. So the process relies on restriction enzymes, which cut the DNA, and also DNA ligase, which joins the DNA. So the enzymes that are responsible for cutting, this DNA of interest, okay, from, let's say, the human DNA, identifying and cutting it is known as, or they are known as, restriction enzymes. These are also known as the scissors of molecular genetics. So restriction enzymes are used to cut DNA. So after this DNA has been cut, so now we need to paste it into the plasmid. So DNA ligase is now the enzyme which is responsible for joining, or in simple terms, you can say for binding our DNA into the plasmid DNA. So they integrate fragments into cloning vectors, typically plasmids or viral DNA, to yield recombinant DNA. So you need restriction enzyme to catch your DNA of interest. In this DNA of interest, for it to be pasted and joined on the plasmid DNA, you need DNA ligase, so that you can come up with now a product which is recombinant DNA. Then after that, so the plasmid is then inserted into the bacteria. So this is telling you that for us to paste our DNA, the plasmid has to be isolated from the bacteria. Then so after that, after insertion of our DNA of interest into the plasmid is then inserted back into the bacteria. Now, you have a number of bacteria. So there's bacteria that is going to take our plasmid, and then there'll be bacteria that will not take the plasmid that contain our DNA of interest. Then after that, there is what we call the selection process. So what is the selection process? We want to select which of these microorganisms have taken our DNA of interest, have taken the plasmids which have our DNA of interest, and which microorganisms have not taken our DNA of interest. So the use of antibiotic selection to identify the bacteria that took up the plasmid is performed at this stage. Then after that, so we're saying number 4, we're saying you grow up lots of plasmid. So how are we growing lots of plasmid? As you culture the bacteria, they begin to replicate. As they replicate, they make copies or they make clones of your plasmid, giving you many copies of your DNA of interest. So you grow up loads of plasmid carrying bacteria and use them as factories to make proteins. Then after that, you can harvest the protein from the bacteria and purify it. Then the last step, or step number 5, is screening and identifying host cell colonies containing recombinant DNA. So this is after you have grown them, and then you identify what are these colonies that are carrying recombinant DNA that you can now okay, so let's look at these steps in detail. Okay, so let's begin with step number 1. Step number 1 is identification and isolation of the DNA of interest. So we're saying A prerequisite step to any recombinant DNA work is the initial isolation of DNA or rRNA from the source material, which can be microbial, plant, animal, or viral. So let's begin. How do we isolate the DNA of interest? How do we isolate this DNA of interest? For example, let's go with the DNA that codes for insulin. So initial revelation of the nucleic acid by disruption of the cell wall present and also end of the cellular plasma membrane, followed by selective precipitation and often chromatography. So we are saying, first step, where do we find DNA? Where are we going to find this DNA? This DNA is found in the nucleus. Where is the nucleus? The nucleus is inside the cells. So the first step in the initial isolation of this DNA, there has to be a revelation or release of the nucleic acid by disrupting the cell wall, and also the cell plasma membranes, followed by selective precipitation and often chromatography. So vigorous methods can result in physical shear or fragmentation of long DNA. Thus, handling is important. So as you are handling, also as you are disrupting the cell wall, disrupting the cell membrane so that you can get to the nucleus, and also like get to the nucleus or the nucleic where you have access to the nucleic acid, or let's say the human DNA. It is important to handle these processes with so much caution, because it can result in either fragmentation. It can result in fragmentation of long strands of DNA. So the gentlest method of cell lysis usually involves incubation with cell wall degrading enzymes. So as we begin, how can we isolate? How do we get to the nucleus? So first of all, we get this. So these cells are going to be incubated with cell wall degrading enzymes. This is a much more gentle method. There isn't a lot of physical shear, which could result in fragmentation of DNA. So get your cells. So your cells are incubated. With cell wall degrading enzymes, and the addition of a detergent will solubilize the plasma membrane. So you have the cell wall. So you use cell wall degrading enzymes, to break down the cell wall. And then the next barrier is the cell membrane. So you can addition of detergents can solubilize the plasma membrane, and hence getting you or leading you to leading to the accession of the nucleic acids. And then now there's purification of DNA. Now that we have gotten to the DNA, there's need for purification of DNA. Normally, it involves solvent-based extraction or precipitation. Okay, now, if DNA is required, then the rRNA present may now be removed by the addition of enzyme ribonuclease, which selectively degrades DNA. What are we talking about? So when you get to the nucleic acids, you have DNA, you also have rNA. So if our interest is the DNA, so since we are interested in the DNA, we need to get rid of the rNA, or we need to remove the rNA by adding enzyme ribonuclease, because these can degrade, or they can selectively degrade rNA. So that we can have a more purified form of our DNA. Then, if eukaryotic mRNA is required, then affinity-based purification may be undertaken using an oligo-DT column. Okay, so we're saying that two approaches have been developed to sequence DNA. So there's the Maxam-Gilbert chemical sequencing method, and also the Sanger-Coulson enzymatic sequencing method. Now, both direct genomic DNA and mRNA, they have a starting point for coding of protein of interest. However, if we're going to use mRNA to code for proteins, if we're going to use mRNA in recombinant DNA technology, then it has to be reverse transcribed. Why? Because mRNA is a single-stranded DNA. Okay, So let's look at the steps that are involved in protein synthesis so that we can understand as we go by how, why, and how this happens. Okay, so we are saying that we have DNA. So after we isolate our DNA, for instance, so we know that DNA, it's just like so we're saying DNA, these are the genes, or let's say a group of genes that code for a specific protein. So DNA can undergo transcription. So it undergoes transcription into RNA, which is normally the messenger RNA, and the RNA is translated into proteins. So how can this DNA that we isolate from the human being, put in the plasmid, how can we make proteins from it? So we know that from DNA can be transcribed into RNA, which is messenger RNA. The messenger RNA is then translated into the protein. So, for example, this is the DNA. If this is the DNA that codes for insulin, it can be transcribed into messenger RNA. Then this messenger RNA is translated into the protein, which is the protein insulin. Then, as we had said earlier, if you're using mRNA, for instance, in the process of recombinant DNA technology, because it's single-stranded, so RNA, it has to undergo reverse transcription into DNA. Then the whole process can begin. Now where DNA is transcribed into RNA, then RNA translated into that specific protein. So after identification and isolation of the DNA, how did we identify? So we identify using restriction enzymes and cut using restriction enzymes, and then we paste this DNA of interest into the plasmid using the enzyme DNA ligase. So let's look into step number 2, which is the cutting and pasting of DNA. Now, how can pieces of DNA from different sources be joined? So a common method uses 2 types of enzymes, which is restriction enzymes for cutting and DNA ligase for joining. A restriction enzyme is a DNA cutting enzyme that recognizes specific target sequence and cuts DNA. So the restriction enzymes have the ability to recognize a specific target sequence. For example, say this is a sequence that codes, let's say a sequence that codes for human insulin. They have the ability to identify it and cut it. Okay, so we're saying they have the ability to recognize a specific target sequence and cut DNA into 2 pieces at or near the site. Many restriction enzymes produce the cut ends with short single-stranded overhangs. So if 2 molecules have matching overhangs, they can best pair and stick together. However, they won't combine to form an unbroken DNA molecule until they are joined by DNA ligase. Yes, we're saying the pieces of DNA that are cut by the restriction enzyme, they are yes, they can be complementary. Yes, they can have sticky ends. But for them to make an unbroken strand of DNA, they need to be joined and sealed by DNA ligase. So DNA ligase is also known as the glue of molecular genetics, just like restriction enzyme is also known as the scissors of molecular genetics. So the initial fragmentation of genomic DNA is undertaken using enzymes known as restriction enzymes. Okay, then these enzymes, one, they recognize, they bind, and they cut DNA sequences which exhibit a defined base sequence. So for example, they recognize a base sequence that codes for insulin, they bind to that region, and then they cut that DNA. So we have over 800 restriction endonucleases that have been identified. So restriction enzymes are also known as restriction endonucleases. Okay, so let's look at this example so that we can understand. So we have this piece of DNA there. So we know that the bases G, so this is guanine, cytosine, adenine, and all that. So we know that G goes with C, A, T, so GCAT, and just like that. So let's say we have a restriction enzyme. So restriction enzymes, they have the ability to identify a sequence that codes for a specific protein, so a specific sequence on DNA. They identify, they bind, and then they cut to produce fragments that have single-stranded overhangs, which can then bind to another double-stranded DNA molecule. So, for example, if you look, if you look at the regions where there are arrows here, where they have identified, so this is the region between G and A, GA, and AG. So it is cut at G on the upper end here. Then it also cuts between A and G at the lower end, producing overhangs, okay, or sticky ends. And then next we're saying fragments with matching overhangs or base pairs stick together. So if this DNA, if this piece of DNA that has been cut, so if this piece of DNA that has been cut is going to be joined to a plasmid, for instance, it's going to be joined to a plasmid, which is another DNA molecule, they need to have complementary ends so that they can pair. So as you can see from the other end, so where we have the G, so down it will pair with the down it's going to pair with the C, just like that. And then so the overhang, so where there's A, for example, so this strand A will pair with the T, so the A here will have to pair with a complementary T strand. Now for this to be joined, for this strand, so this is the strand of DNA. So for this strand of DNA to be joined, you need DNA ligase, which then seals them. So DNA ligase seals the gaps. So our goal in cloning is to insert a target gene, for example, human insulin, into a plasmid using a carefully chosen restriction enzyme. Okay, so one, we're saying we digest the plasmid, which has a single cut site. So here is one point to remember. The plasmid, the restriction enzyme which is used to cut the DNA of interest, it is also the same restriction enzyme that is used to cut open the plasmid so that it can make complementary overhangs with the DNA of a plasmid that is coming in the area where it has been cut, so that it can make complementary ends. So we're saying we digest the plasmid, which has a single cut site, then the target gene fragment, which also has a cut site near each end. We then combine the fragments with DNA ligase, which links them to make a recombinant plasmid containing the gene. Okay, overall, what are we saying? This is the plasmid. So a full plasmid, as you can see, it's not cut. So this is the plasmid from bacteria, okay, or let's say from E. coli. So the plasmid is incubated, or the plasmid is incubated with restriction enzyme. Then the restriction enzyme, okay, so it will also identify an area. So it identifies a specific site where it can cut, which will be complementary with the target gene that is coming. So the same restriction enzymes which cuts open the plasmid is the same one that is used to cut the DNA of interest from the normal human genome. So you cut open the plasmid so that you can make complementary ends using restriction enzyme. Restriction enzyme, again, it's the same one which is used to cut the gene of interest, for example, insulin, which can now bind in this region. Why? Because they are complementary ends. So for example, if this end was A on the target gene, the other end here will be T, for instance. If this end here is G, then the other end this side will be C. So they make complementary overhangs. Then they are joined by ligase to form recombinant plasmid. So DNA has been recombined, and hence recombinant DNA technology. The third step is bacterial transformation and selection. Now, so remember we're saying plasmids and other DNA can be introduced into bacteria, such as the harmless E. coli. So E. coli is the most commonly used bacteria in the production of most of these biological products. So we're saying in this process is called transformation. So what are we saying? So now that we have the plasmid, so remember the plasmid was isolated from the bacteria so that we can paste our DNA of interest. After making this recombinant plasmid, it is then reintroduced into the bacteria, where after bacteria begin to replicate, now we can make copies of our DNA of interest. So plasmids are introduced into the bacteria, such as E. coli. And this process is called transformation. So the reintroduction of a recombinant plasmid into the host cell, which is the bacteria, is known as transformation. During transformation, specially prepared bacterial cells are given a shock. So we are saying, how then do these bacteria take up these plasmids that contain our DNA of interest? So they are given shock. Okay, so bacterial cells are given a shock, such as a high temperature, that encourages them to take up foreign DNA. So we're saying you're going to have this solution where you have bacteria and also the plasmid DNA. So this solution is heated to a given temperature, which encourages, or so the shocking of the bacterial cells, such that it encourages them to take up the foreign DNA, or the plasmids that contain the foreign DNA. Then these engineered vectors are introduced into the cells. So engineered vectors, we're referring to the plasmids. So they are introduced into the host cells. This can be achieved by incubating the host cells together with the vector in ideal solvent. So you get an ideal solvent, get your E. coli or bacteria that are supposed to take up these plasmids. So they are okay. So I'm saying okay. So this is achieved by incubating the host cells together with the vector in an ideal solvent, which is then heated as a way to introduce the shock, which encourages uptake of the foreign DNA or the recombinant plasmids. So processes to facilitate entry of vectors into cells are conducted. This includes application of rapid temperature changes. This could be associated with improved kinases. Okay, so the use of heat shock, it's thought that the heat shock changes the fluidity of the membrane and causes pores, to form, making it easier for DNA to get across or enter the cell. So the theory behind heating the solvent that contain the host cells or bacteria is that it increases the fluidity, or let's say it increases the porosity. In simple terms, increases the porosity of the bacterial cell wall or the membranes, such that it's easier for them to take up these plasmids. So host cells that have successfully integrated recombinant DNA are cultured and grown. Strategies to identify cells housing the plasmids are applied. So, of course, there'll be those bacteria that take up the plasmids, and then there'll be those that do not take up the plasmids. So this is where we're saying strategies to identify the cells housing our recombinant plasmids are applied. Then host cells that fail to take up the plasmids do not express the DNA are killed. So after the ones that took up, so after identification of the cells that took up our plasmids, there's a technique that is applied to destroy or to kill all cells that do not contain the DNA of interest, so that we can just now grow or clone the cells that have our DNA of interest. Then modes of separating host cells that have not taken up the plasmids are carried out. Okay, so let's see what happens here. Now remember, we have ligation products. So what do we have here? So remember, we have the plasmids. So these are the plasmids that have taken our DNA of interest. So plasmids that have been ligated with our DNA of interest. And then you get bacteria in a suitable solvent. And then to this solvent, we add, so to this solvent, let me just get, okay, so here, let me use red. Okay, so here we're saying after we've made the recombinant plasmids, so these are the plasmids that are carrying our DNA of interest. They need to be reintroduced into the bacteria. So the bacteria is put in a suitable solvent, and then our plasmids that contain the gated product. So these plasmids that contain our DNA of interest are then incubated together with the bacteria. So after they are added to these bacteria, to this bacteria containing solution, then this solution is heated. So there's heat shock. So we're saying heat shock is believed, or heating is believed to increase the fluidity or the porosity of the either, let's say, the cell wall or the plasma membranes of these bacteria, such that it encourages uptake of these plasmids. So as you can see, now after heat shock, after heat shock, these individual so you have a number of bacteria. So you have bacteria that have taken your plasmid here, and then you have bacteria that have not taken your plasmids. For example, you have bacteria that have not taken your plasmids. These are still in one container. So you can have those that take up your plasmid. So this is your original plasmid. After heat shock, some will take the plasmid, some will not take the plasmid. That's why we're saying techniques to identify which bacteria have not taken the plasmids are applied, so that we can only remain with bacteria that have taken, or that have been transformed, that have taken our DNA of interest, so that then this can be cultured. Okay, so let's look at the next step, which is selection. Now, a plasmid typically contains an antibiotic-resistant gene. So I'll keep this very simple for the purposes of understanding. So in the plasmid, a plasmid also contains what we call an antibiotic-resistant gene. So a plasmid typically contains an antibiotic-resistant gene, which allows bacteria to survive in the presence of a specific antibiotic. Thus, bacteria that took up the plasmid can be selected on the nutrient plates containing the antibiotic. Bacteria without the plasmid will die, while bacteria carrying a plasmid will live and reproduce. Each surviving bacterium will give rise to small dot-like groups, or a colony of identical bacteria that all carry the plasmid. What are we talking about? So remember, so all the recombinant plasmids that contain our DNA of interest, they also contain an antibiotic-resistant gene. Our recombinant plasmids, apart from them carrying our DNA of interest, they also carry an antibiotic-resistant gene. What is an antibiotic-resistant gene? Such that every plasmid of ours, so which contains our DNA of interest, it should also carry an antibiotic-resistant gene, such that when they are cultured, so when an antibiotic is added to this colony, for example. So what is a colony? So remember in the previous slide, there were those let's say there was bacteria that took up our recombinant plasmid, and then there's bacteria that did not take up our recombinant plasmid. So every bacteria that has taken up our recombinant DNA, its plasmid will also have an antibiotic-resistant gene, such that in the presence of this antibiotic, all microorganisms, or all let's say all host cells that are carrying a plasmid DNA of interest will not die, or will not be killed. Why? Because they are resistant to the effects of that antibiotic. So it means every other bacteria without the plasmid. So that means if they do not have this plasmid which carries our DNA of interest, or without a plasmid, they do not carry the antibiotic resistant gene. So in the presence of this antibiotic, they will die. So in this colony, so what is this colony? So we're saying a group of cells that have replicated. We have cells that have taken the plasmid, the recombinant plasmid, we have cells that have not taken the recombinant plasmid. So all those cells that have taken the recombinant plasmid, they're not going to be killed in the presence of an antibiotic. So antibiotic is added to this colon. They will survive. Why? Because they contain antibiotic resistant gene. All those bacteria that have not taken the plasmid will die. Why? They do not contain a plasmid with an antibiotic resistant gene. So this is where we're saying bacteria without plasmid will die, while bacteria carrying a plasmid will live and reproduce. So this is how we select. So this is how we select the bacteria that have taken our DNA of interest, and then they are later cultured. So not all colonies will necessarily. Okay, so we're saying not all colonies will necessarily contain the right plasmid. That's because during ligation, DNA fragments don't always get pasted in exactly the way we intend. Instead, we must collect DNA from several colonies and see whether each one contains the right plasmids. Okay, so methods like restriction enzyme digestion and PCR, which is polymerase chain reaction, are commonly used to check the plasmids. Okay, and then we have now protein production. How do we produce proteins? Now that we have these bacteria that have taken our plasmids that contain our DNA of interest, it's just DNA there. How do we make protein? So if you remember the process of protein production from DNA, DNA is transcribed into RNA, and then the RNA can be translated now into that specific protein. So protein production. Now, once we have found a bacterial colony with the right plasmid, we can grow a large culture of plasmid-bearing bacteria. Now that we have only bacteria that have our plasmids carrying the DNA of interest, we can grow them, or we can culture them so that we have colonies, or so that we have, okay, let's just say we have a pool of our gene of interest. So we can grow a large culture of plasmid-bearing bacteria. Then we give the bacteria a chemical signal that instructs them to make the target protein. So we cause these bacteria to express the protein for which the DNA that they are carrying codes for. So by chemically signaling them, then the bacteria serves as miniature factories, churning out large amounts of proteins. So we know that bacteria can replicate. So as they replicate, they make millions and millions and millions of copies of our DNA, which can then be used to express, which can then be chemically signaled to produce large amounts of proteins. For instance, if our plasmid contained the human insulin gene, the bacteria would start transcribing. So we have the DNA that codes for insulin is transcribed. So the bacteria will start transcribing the gene, which will then be transcribed into messenger RNA. Then the RNA will be translated to produce many molecules of the human insulin protein. So yeah, in a diagram, what are we saying? So first we have colony is grown into a large culture. So this is a large culture of bacteria that contain our DNA of interest. So it is chemically signals. So this is indicating of a large amount of colonies that have been of a colony that has been grown, or that has been cultured with increased pool of the human DNA or the gene of interest. The bacteria are induced to express the gene. So the bacteria are chemically induced so that they can express the gene for which they are carrying. What is this gene, for instance? Let's say the human insulin gene. So when the bacteria is chemically signals, or it is chemically induced to express the gene, so what will happen is, remember, this is the plasmid that carries your DNA of interest. So the plasmid, which is the DNA, it undergoes transcription. So it is transcribed into messenger RNA. It is transcribed into messenger RNA, so which is a single-stranded DNA, and then RNA, so which is a single-stranded molecule, and then RNA is then translated into that specific protein, being human insulin. So what do? you have bacteria that are carrying your gene of interest. This bacteria can be caused to express the gene, so they're chemically signaled, and then after that, so the gene of interest, the DNA undergoes transcription to make messenger RNA. The messenger RNA is then transcribed to make these proteins. So DNA of interest makes messenger RNA. Messenger RNA is then now transcribed to make insulin proteins. So remember, the insulin is still inside your bacteria. So once the protein has been produced, the bacterial cells can be split open to release it. So you have this protein. So the protein that you've produced, the bacteria, yes, they have expressed the gene that they are carrying to make the protein, but it's still inside the bacteria. So it means the bacteria has to be lysed, or has to be split open to release it. There are many other proteins and macromolecules floating around in bacteria besides the target protein. So remember, when you lyse open, or when you, in simple terms, so the bacteria is broken, so it's lysed, it's split open so as to release the contents inside. One of the contents that we're interested in is the target protein, or the insulin that has been produced. But remember, there are all other proteins that are inside, that are produced by the bacteria as they replicate. So there are many proteins and other macromolecules floating around in bacteria besides the target protein, for example, insulin. Because of this, the target protein must be purified. The target protein must be purified or separated from other contents of the cells by biochemical techniques. So here, biochemical techniques have to be applied so as to separate the protein of interest that you are interested in from all other proteins and macromolecules. The purified protein can be used for experiments, or in the case of insulin, can be administered to patients. So after that, now that we've lysed the bacteria, then we can use biochemical techniques to identify, or to target, and also purify our protein of interest. Then after that, these various proteins can be used either in experiments. In our case, if it's insulin, then it can be used in the treatment of insulin-dependent diabetes. So the methods of purification we discussed later. And then we look at uses of DNA cloning, or recombinant DNA. Why is it important? Or what are some of the uses? What are some of the applications of DNA cloning, or the general recombinant DNA technology? Process one, so it can be used in manufacturing of biopharmaceuticals. What are biopharmaceuticals? So pharmaceutical products that manufactured using biotechnology or recombinant DNA technology, or this process, are known as biopharmaceuticals. So DNA cloning can be used to make human proteins with biomedical applications, such as insulin, as mentioned above. Other examples of recombinant proteins include human growth hormone, which is given to patients who are unable to synthesize the hormone, and also the TPR, which is the tissue plasminogen activator, which is used to treat strokes and prevent blood clots. Recombinant proteins like these are often made in bacteria. So making biopharmaceuticals or biologicals. What are these biologicals? For example, insulin, as it's shown here. So for example, we have insulin, for example, growth hormone. It's also used to make tissue plasminogen activators and all that. Apart from formation of biopharmaceuticals, it can also be used in gene therapy. What is gene therapy? So gene therapy. So therapy is treatment. So we want to treat genetic disorders, treatment or correction of genetic disorders. So in some genetic disorders, patients lack the functional form of a particular gene. So we're saying gene therapy attempts to provide a normal copy of the gene to the cells of a patient's body. So in gene therapy, or let's say in genetic disorders, the genes are coded for a specific protein, or let's say for a specific characteristics. Normally they are dysfunctional. So we're saying a right copy, or a normal copy of DNA can be made, okay, outside the body, and then introduced into the cells of the patient. So you make a normal copy and hence DNA cloning. So make a normal copy of this gene, then it's introduced into patient's cells. For example, DNA cloning was used to build plasmids containing the normal version of the gene that's non-functional in cystic fibrosis. When the plasmids were delivered to the lungs of cystic fibrosis patients, lung function deteriorated less quickly. So it can be used in biopharmaceuticals, it can be used to correct gene disorders, then it can also be used in gene analysis. So in basic research labs, biologists often use DNA cloning to build artificial recombinant versions of genes that help them understand how normal genes in normal organisms work. So in gene analysis, you want to make copies, artificial copies of DNA, so that you can help, so that they can assist or they can aid in understanding how normal genes in an organism functions.